Hello boys, welcome back to another session of video tutorials, and this session is meant for class 12th biology students, and this is for 3rd September 2020. Our memory helps us in tracing out what we have been discussing and arriving at, plus it links from the past to the present. In our previous session we were talking about rDNA technology, various steps involved in rDNA technology. Just to help your memory, we were talking about the following steps. Isolation of genetic material, that is DNA. In order to cut the DNA with restriction enzymes, it needs to be in pure form, free from other macromolecules. This can be achieved by treating the bacterial cells, plant or animal tissue with enzymes such as lysozyme, cellulase and chitinase. The RNA can be removed by treatment with ribonuclease, whereas proteins can be removed by treatment with proteases. The purified DNA finally precipitates out after addition of chilled ethanol. This is seen as collection of threads in suspension. This experiment, of course, we will be doing this in our lab. And individually you will be extracting DNA from both plant sources as well as animal sources. And step number 2 in recombinant DNA technology is cutting of DNA at specific locations. We were talking about the function of our restriction endonucleases. So restriction enzyme digestions are performed by incubating purified DNA molecules with restriction enzyme and the optimal conditions for the specific enzyme. Agarose Gel electrophoresis is employed to check the progression of restriction enzyme digestion. So gene of interest, yesterday we were talking about that. From the source DNA and the cut vector with space are mixed and ligase is added. This results in the preparation of rDNA, recombinant DNA. If required, gene of interest is amplified using PCR. And third step we were talking is insertion of rDNA into suitable host cell or organism. So with that in mind, we were going to discuss about the gel electrophoresis, which is very important. And this gel electrophoresis help us in making the gene of interest in more numbers. We can separate the DNA fragments through gel electrophoresis. So in this first step, action of restriction endonuclease results fragments of DNA as we discussed, and it is separated by a process called gel electrophoresis. And much depends on negative nature of DNA. External electrical field is applied to the medium and DNA move to anode. Medium or matrix used is agarose gel. You must remember this word, agarose gel. Many times it will be asked as 1 mark question in CBSE. And this is natural polymer and extracted from seaweed. This also you must remember, it's a natural polymer extracted from seaweed. And medium acts as sieve and separate DNA according to their sizes. This is the importance of agarose gel. It acts like a sieve, like your mummy separates, using that small instrument in your kitchen. Sieve one separates the bigger particles from smaller particles in rava. So the smaller fragments further will be separated. So separated DNA parts stained with ethidium bromide. This is a stain, okay? Ethidium bromide is used, and after applying the stain, the DNA fragments are exposed to ultraviolet radiation, and because of this ethidium bromide, bright orange colored bands of DNA will be appeared in the gel. That is the importance of ethidium bromide. And separated bands of DNA cut out from agarose gel, extracted from gel piece. This process is called elution. So elution is separation of the DNA fragments from the medium of Agarose Gel. Purified DNA fragments constructing recombinant DNA by joining them with cloning vector. So once we do the elution process, then purification of those DNA fragments to be done. And once the DNA fragments are purified, we use it for construction of rDNA. And we can see how exactly this gel electrophoresis machine appears. This is how the gel So you've got a cathode and anode, what we call sides, cathode side and anode side. And you've got a gel, that Agarose gel. So it's a mixture of DNA molecules of different sizes. We pour in that and make the entire thing to be under the influence of electricity. That's why we are saying electrophoresis, separation of DNA fragments based on size. And you can see in the second diagram wherein the DNA fragments are separated based on size. So one side you are seeing longer molecules and other side you are seeing shorter molecules. And of course we said once the DNA fragments are separated based on size through the electrophoresis, we need to do staining. Staining means coloring, and that staining is ethidium bromide. And once it is stained, you can see the orange color here, orange color fragments. And this is exposed to UV radiation. The cutting out DNA fragments are exposed to DNA fragments. So basically, electrophoresis is a technique of separation of charged molecules under the influence of an electric field so that they migrate in the direction of electrode bearing the opposite charges. That's what we said, anode and cathode. Cathode minus, anode plus. And this happens through a medium or matrix. What is the medium? Agarose gel. The most commonly used matrix is agarose, which is polysaccharide extracted from seaweeds. Next, DNA fragments separate according to size through the pores of agarose gel. That is the reason why we are using agarose gel. It acts like a sieve, natural sieve, and it's a natural polymer. And then the separated DNA fragments can be seen only after staining the DNA with a compound known as ethidium bromide, ETBR, ethidium bromide. And this is followed by exposure to UV radiation as bright orange colored bands, as you are seeing. And the separated bands of DNA are cut out from the agarose gel and extracted from the gel piece. This step is called elution. So here you have to remember two things. What is gel electrophoresis? One. Second, what is elution? So gel electrophoresis is a technique of separation of charged molecules under the influence of an electric field so that they migrate in the direction of electrode bearing the opposite charge. You can see anode and cathode through a medium or matrix called agarose gel. And elution is a step that helps in separating the DNA bands from the agarose gel medium and are extracted from the gel pieces. This is the importance of gel electrophoresis. And once that is done, then we go for the cloning vectors. A cloning vector is a small piece of DNA taken from a virus or a plasmid of bacterium that can be stably maintained in an organism and into which a foreign DNA fragment can be inserted for cloning purposes. That is what we call cloning vectors. The vectors are DNA molecules that can carry a foreign DNA segment and replicate inside the host cell. That is the purpose. The common vectors may be plasmids, bacteriophages, cosmids, and YACs. The expansion is yeast artificial chromosomes, and BACs, bacterial artificial chromosomes, and transposons and viruses. These are the common vectors. Out of these vectors, plasmid vectors and bacteriophage vectors are commonly used. What are the salient features of cloning vectors? They have ability to replicate within bacterial cell independent of chromosomal DNA. High copy number per cell. Plasmids somewhere 15 to 100 copies per cell. Third, alien DNA when integrated with vector can multiply equal to copy number of vector. Vectors can be engineered for easy linking of foreign DNA and selection of recombinant and non-recombinants. So out of these, as I said, plasmids are extra-chromosomal, self-replicating, circular, double-stranded DNA molecules found naturally in many bacteria and also in some yeast. That's how we pick up the bacteria and viruses. Plasmids are usually not essential for normal cell growth and division, but they often confer some traits to the host organism, like resistance to certain antibiotics. So the most widely used, versatile, easily manipulated vector is PBR322. PBR322 is an ideal plasmid vector. So we will be talking about that PBR322 separately in our next session, and also we will be talking about gene cloning with different vectors. Like we are going to talk about bacteria, like this is the bacterium host cell and the plasmid DNA is there. We are taking out that, and to that vector DNA we are adding foreign gene, so that becomes recombinant DNA molecule, and that is made to enter into the host cell, and the host cell undergoes either binary fission or multiple fission. As the host cell multiplies, normally bacteria, the rDNA also multiplies. This is how we increase the numbers of genes of interest. Similarly, if the vector is virus, so you got a viral DNA, and to that viral DNA we are adding this foreign gene or gene of interest. You get a recombinant DNA molecule, and this recombinant DNA is inserted into the host cell package into mature virus by transfection. And once this enters the host cell, the viral DNA or recombinant DNA keeps on multiplying it, and finally there will be cell lysis. The host cell will be broken and recombinant DNA molecules are released in plenty. So this is how we select the normal cloning vectors, or we say vehicle DNA is either bacteria or virus. We further talk about that tomorrow, the famous one PBR322. Until then,